Hello, in this video we look at the Nobel Prize in Chemistry for the year 1902, awarded to Emil Fischer. This is the first video on this. The second video will deal with his lecture. This is more general. And why did he was awarded the prize? For his work on sugar and purine synthesis. This is the shortest way to saying that, coming from Wikipedia, by the way. Here you see what he was working on. He was working on sugars, and you see here left you see a sugar molecule of the glucose kind, and here you see a variety of that, the fructose kind. In here they are connected, so you have a disaccharide, two sugar molecules connected. If you split them, you get two monosaccharides. Okay, first from the official side, something about his work, why he got the prize. Carbohydrates, including sugar, are crucial to the biological processes of living organisms. In 1877, Emil Fischer produced phenylhydrazine, a base that helped reveal sugar molecule structures. Fischer was able to produce several sugars artificially and to map the structures of their molecules. Another group of substances with important biological functions is nitrogenous compounds or purines. Fischer showed that purines form the caffeine found in coffee and its equivalent in tea and in cacao. This is the official reason why he got the prize, and you can see this is come from the official Nobel Prize site, and you can go there by following the link. You again see two sugar molecules. You can see these are monosaccharides, glucose and glucose, but they are different. You see D and L. If you look very good at the structural formula, you see that the H and the OH group in this are symmetrical with this one. Here the AOH is on the left side and the H on the right side, and here it's the other way around. And this is also true for some of the other groups, as you can see here. You know, this one is also a mirror image, this is a mirror image, and this is a mirror image. But these parts of the molecule and this part is not. Yeah? So two different forms of the same molecule, the same different structure, but the same content. The work, however, this is from the biological part of the Nobel Prize. The work, however, on which Fischer's fame chiefly rests, this is there's already a part before it, which I have skipped, was his studies of the purines and sugars. This work, carried out between 1882 and 1906, showed that various substances such as adenine, xanthine, in vegetable substances, caffeine, and in animal excrete, urine acid and guanine all belong to one homogeneous family and could be derived from another, and that they correspond to different hydroxyl and amino derivatives of the same fundamental system formed by a bicyclic nitrogenous structure into which characteristic urea groups entered. So they're all part of the same family. They have more or less the same structure, and they can be derived from another, as I said. This parent substance he called this parent substance he called purine in 1884, and he synthesized it in 1889. Numerous artificial derivatives, more or less analogous to the naturally occurring substances, came from his laboratory between 1882 and 1896, and between 1899 and 1908, Fischer made his great contribution to the knowledge of the proteins. That's another group of chemical compounds, of course. He sought methods of separating and identifying the individual amino acids, discovering a new type, the cyclic amino acids, proline and oxyproline. He also studied the synthesis of proteins, and he was able to establish the type of bond that would connect them together in chains, namely the peptide bond, and by means of this he obtained the dipeptides and later tripeptides and polypeptides. In 1901 he discovered, in collaboration with Fourneau, the synthesis of the dipeptide glycine and in that year he also published his work on the hydrolysis of casein. Amino acids occurring in nature were prepared in the laboratory and new ones were discovered. His synthesis of the oligopeptides culminated in an octodecapeptide which had many characteristics of natural proteins. If you want more to know about those compounds, you can of course look at Wikipedia or Google them. Of course, I'm not gonna speak about them all. Some of them will come back later in the speeches, the award speech and the Nobel Prize speech. All this information, of course, again is coming from the official site, and you can see it's only part of it. He did more than this, but I think this was the most important part of his contribution to science. Here again you see purines was mentioned lately, and you can see that all these compounds are family, of course. They all have a basic structure which is more or less the same, and different groups are added to the ring structure, the double ring structure you can see here. Here you have a 6-part ring connected to a 5-part ring. They have all that in common. Then the award ceremony was preached by Professor A.G. Thiel, probably pronounced wrong. Apart from the protein substances, there is no group of carbon compounds more important for organic life than the carbohydrates. For that reason, the carbohydrates, and in particular the sugars, have been the object of countless studies. Owing to the nature of these substances, their study was fraught with great difficulty and until a few years ago appeared an impossible task. On the basis of this discovery of hydrazine derivatives, Fischer succeeded in finding a brilliant solution to the problem. Fischer not only reproduced synthetically natural grape and fruit sugars, but also some 30 other sugars and an abundance of closely related compounds. Naturally occurring simple sugars, only those with 5 or 6 carbon atoms have been found. Fischer synthesized a continuous series containing from 2 to 9 carbon atoms. He also added to the achievement of organic synthesis this group which is so extraordinarily important for vegetable physiology. These students have been significant especially for the theory of spatial arrangements of atoms as elaborated by Van Hoff and Nobel, previous Nobel Prize in chemistry. Fischer succeeded in determining fully not only the way in which the atoms are bound in the atomic complexes, the molecules, but also the position of the atoms relative to another, or in other words, the stereometric configuration of the molecules. Since the degree of complexity here is great, our conception of atomic grouping has attained such a degree of certainty through these studies that we can be convinced that the conception will never be substantially weakened, even were the conception of the nature of atoms and particularly of valences to undergo radical changes in the future. These studies have been no less important for physiology. One of the most superb processes occurring in nature is the formation of carbohydrates in the green parts of plants. As early as 1870, A. von Baeyer, also Nobel Prize winner later, formulated the hypothesis that carbon dioxide and water are reduced to formaldehyde or formalin in cells which contain chlorophyll, and that is immediately condensed to form sugar. And Fischer has now succeeded in preparing from formaldehyde both grape and fruit sugar, which occur universally in plants, and beyond that constitute the parent substances for the other carbohydrates. The hypothesis in question has thereby acquired experimental confirmation of obvious value. The sugar syntheses are furthermore associated with inorganic carbon, hydrogen and oxygen via formaldehyde. Enzyme actions, which have such a profound bearing on the vital processes, appear in altogether new perspectives as a result of these studies. It was found that the synthetic sugars with 3 and 9 carbon atoms are converted into alcohol and carbon dioxide by yeast as readily as grape sugar, which contains 6 carbon atoms, but that after slight modification in the stereometric configuration of the molecule, the latter is completely unaffected by the same enzyme. Here we encounter the important discovery that the vital function depends more on the geometrical configuration of the molecule of the nutritive substance than on the composition in other aspects. Fischer determined in a similar sensitivity the so-called asymmetric structure for other enzymes, too, and for the glucosides. Through this observation, molecular asymmetry has gained formerly unsuspected importance. It was found that the enzymes themselves, in common with the most important products of plant life, carbohydrates, proteins, chlorophyll granules, and protoplasm itself, are without exception optically active substances, or else are composed of them, and that all essential chemical transformations in the organism depend on asymmetry. These sugar syntheses are indeed the very first processes in which the action of enzymes can be verified in detail and with accuracy. For animal life, the nitrogenous substances occurring in the organism are the most important. Protein substances apart, the animal body contains a substantial amount of other nitrogenous substances. The study is of great value for physiological chemistry because they are either products arising from the use of proteins or, together with simple proteins, are contained as components in the most complicated compounds of the organism, the purines, as they are called. Even since 1776, when Scheele found uric acid in urinary calculus, several substances closely related to it, as xanthine, adenine, and guanine, etc., have been detected in animal secretions. The same group additionally includes theobromine, theophylline, and caffeine, which occurs in the vegetable kingdom and constitute the stimulants in our staple beverages, coca, coffee, and tea. Fischer brought order and clarity to this field as well. He demonstrated that all those substances are derivatives of the same parent substance, purine, which he had discovered. Here we see again a lot of monosaccharides, which you can see are all more or less the same, but they are stereometrically different. They have a different shape, or even have the same carbon and hydrogen and oxygen molecules. He successfully prepared them for another and from simpler constituents in such a way here too the synthesized chain goes back into organic carbon, hydrogen and oxygen. And besides these, he prepared a large number of new closely related substances, so that the purine derivatives studied by Fischer must now number about 150. The intrinsic composition of each has been fully determined. For physiology, the experimental proof that uric acids and the xanthines originate from one and the same parent form is of the greatest importance, as it has proved the strongest support for the modern theory of the formation of uric acid from the nuclear proteins of the cell nucleus and from the purines contained therein. A certain representatives of the groups, caffeine and theobromine, are not solely esteemed in beverage form, but have also been used medicinally down the ages. It is reasonable to expect that several of the new purine derivatives will prove to have a medicinal value. Well, the chemical industry too has already made initial attempts to work out this for its own purposes. Fischer's synthesis of these substances, which are so greatly appreciated in daily life. The specific type of research which characterized organic chemistry during the final decennia of the century that has just closed attained its zenith of development, and from its finest form in Fischer's study of sugar and purine. From the experimental point of view, they are unsurpassed. Again, coming from the Nobel Prize site, here you have the link, and even more information you can also find out if you follow this link, the so-called speed read. Here again you see glucoses, and you can see that they are all more or less the same. It's a study in itself. Well, thank you for watching. In the next video we'll go into the Nobel Prize lecture given by Emil Fischer, of course expanding on all this. Thank you for watching, and maybe till another time.